Alright, so I thought I would go through and show you the answers to these quiz questions, as I was kind of hoping you would do these. And this is important, so I'll toss my name on here. These are the basic ratios of Mendelian genetics, and so you really need to know the names of all these crosses. And because you've had this stuff before, it's time to really solidify this in your brain if you haven't done that yet. So you just need to know what these terms mean. So a monohybrid cross is always that. And we need to start, we'll do this way so we can get 1, 2, this thing doesn't like dots. Two ways to get this, one way to get that, or if we, so just remember that at least, and then also you'll also need that. And then this is, because these guys give you the same phenotype, we get 3 of this dominant phenotype and 1 of the recessive phenotype. So you should know immediately, monohybrid cross, 1 to 2 to 1 and 3 to 1. Test cross of a monohybrid. That's a monohybrid. The word hybrid means it's a mixture of two things, and this is, so we've crossed two things together to make this hybrid. So a hybrid is heterozygous. A test cross is always crossed to homozygous recessive for whatever genes are involved. So then the genotypic ratio, we can put in the full genotypic ratio here because there's only 2 things. And so we get one of those and one of those. The phenotypic ratio in this case is the same as the genotypic ratio. So again, we're just using these as designations for the phenotype. Dihybrid cross is always this. Now here the full genotype is too much, so during the quiz I said to do the abbreviated genotypes. We could write all those out, but there's 16 different ones. So that you would have to write out the, either do the Punnett square and write in all the boxes and count them all up, or there's a quicker way to do it by multiplying probabilities, but I never really see that it's all that important to write out the full genotypes. So we'll just do the abbreviated. And I guess we'll go the long way here. So 9, and this is going to be really big for the second part of chapter 5, when we're looking at crosses involving 2 genes, and so you just need to get used to writing this down. And it's very specific for this, and that's a dihybrid cross. 2 dihybrids crossed together. Then your phenotypic ratio is 9 of the dominant with both dominant phenotypes, 3 of the dominant A phenotype and the recessive B phenotype, 3 recessive A, dominant B, 1 recessive of each. So those are, a little bit abstract symbols, but if we were using an actual cross, this might be yellow and smooth, yellow and wrinkled, green and smooth, green and wrinkled, whatever the phenotypes are. Test cross of a dihybrid. Dihybrid always means this. A dihybrid cross, and that's why I clarified it here, up top, a dihybrid cross is 2 heterozygous things crossed together. Here, a test cross is always, 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 always that. If there's 3 genes, you'd put 2 little c's, but it's always recessive for both, for all the genes involved. And so, full genotypic ratio. Now, this is where we can say we have to get, we always have to get an A and a B. If we get a big A, we can get a big B or a little b. If we get a little a, we can get a big B or a little b. And so, from this parent here, we'll get a big A and a big B. And then from this parent over here, will always get a little a and a little b. And so that's the first genotype. From this one, we can get a big A and a little b. So you could do this in a Punnett square. I don't really mind Punnett squares that much for just simple crosses. But from here, we always get a little a and a little b. From over here, now we can get a little a and a big B. And from over here, we always get a little a and a little b. And then the last one, we'll get little a and little b from here. So these things, and then over here, we'll get little a, little b, because that's the only thing we can get. So these things that I'm putting in, those would be the gametes from the dihybrid parent. And then our phenotype, so from this parent, what we're doing is saying we're getting these gametes. And then every time we get those gametes, they can only go with that. Now, a couple people did this and then put that down there 4 times. If you want to, if it's the same thing over and over again, you can just put it down once. Then you just fill these in. Big A, but you get a little a. Big B, but you get a little b. But we also want to just memorize this. And when we take tests and stuff, we might not have time to do Punnett squares every time we try to figure something out. Phenotypic ratio. This is going to have the big A phenotype and the big B phenotype. Big A, little b, little a, big B, little a, little b. These are 1 to 1 to 1 to 1, and that's because we have independent assortment. This is really important. This type of cross, a test cross of a dihybrid, that's all we'll do once we get to, I think it's chapter 6, when we do genetic mapping. We're going to do these things all the time. And we're going to be comparing what we get with independent assortment with what we get when we don't have independent assortment. So we need to really have a solid understanding of what happens with independent assortment. And then drawing chromosomes. So really get used to this. This is in the no surprises, no excuses category. If you know something that you have to absolutely know for a test and you're having trouble understanding it, then it's up to you to figure it out. So you need to draw these things out. I'll explain it as many ways as I can. Go to the study tables. Come to my office, ask your friends, Google it, whatever it takes. All right, so consider an organism with 2 pairs of homologous chromosomes, so 2 pairs, so that means 4 total. The organism is heterozygous for 2 genes, are in different chromosomes, that's important. Anytime you see different chromosomes, then we know independent assortment is going on. And it has a genotype of this. Draw the chromosomes as they would align during metaphase of meiosis 1. Chromosome, centromere, 2 chromatids. I don't really care exactly how you draw your chromosomes as long as they represent the right thing. So here's 1 chromosome, and it has a big A on it. And it is going to pair up with its homologous partner. And that's going to have little a's on it. I did see some people that had this with the little a. These guys always have to be identical. These are sister chromatids. They're identical sister chromatids. Now, I didn't say anything about ignoring crossing over here. Oh, yeah, I did. All right, so good. So no crossing over. So that means, so if you do have crossing over, you can flip these things. But that's why we're ignoring it, because I don't want to confuse the issue. And then these, that's a homologous pair. So those are homologous chromosomes. But let's put the rest of the chromosomes in there first. Scroll down and get a little room. Now, those guys, the big A and the little a chromosomes, need to line up the way they are because alleles segregate. When we divide the cell, the big A and the little a are going to go into different cells. And then, but big B and little b, they have their own chromosome. And so when we have cell division, and so this is meiosis 1, and you don't have to necessarily draw in the spindles, but there they are. So the B's are going to go to, big B's are going to go to the left, big A is going to go to the left, little a is going to go to the right, little b is going to go to the right. What we'll end up with is both of these chromosomes, so it'll kind of split down the middle, and the big A and this whole chromosome and the other one will go to a cell. But we're going to kind of skip over that part because meiosis 2 doesn't really affect the total outcome. So down here, what kinds of gametes? That's all I really asked. I didn't ask you to. I think I might have asked you to draw what the chromosomes look like in a gamete. But first of all, the types of letters, the types of alleles that go into the gamete are a big A and a big B, just because that's the way they're lined up in this particular case. And that's a gamete. So you have to let me know what is actually going into that gamete. And this one would have a little a. I'm just drawing my chromosomes a little bit different here. Little b. So that's two of the types of gametes you can get. Then the way you get your other gametes is in another cell of the same organism. So it also has the same genotype. And we can draw our chromosomes here. And our A, as long as the big A lines up with the little a in a pair like this, you're okay. But if we're going to get the other kinds of gametes, here we had big A going with big B, and you always have to, you can't end up with a gamete with two A's in it. You have to have just one of each letter in the gamete. So that means that the big A can go with, when this cell divides, it's going to go with a B. So you always have an A and a B in there. But in this case, it can line up so that it goes with little b, which means big B has to be over here. And there's got to be a big B coffee joke in there somewhere, but I say big B a lot. Maybe if you studied genetics down at big B, it would stick a little bit more. But a lot of you did awesome on this. It's just kind of tricky. And I think one of the issues is you've done it before, and maybe you've developed some bad habits. And so it's a good time to break those habits and really think about this stuff. So that's what the gamete would look like. Because the big A and the big B are coming from this alignment, and then the little a and the big B will come from this alignment. All right, so that's it. Practice, practice, practice. Do it a bunch of times. These are your gametes. They're equally likely. 1 to 1 to 1 to 1, or 1/4 to 1/4 to 1/4 to 1/4. We're correlating how the chromosomes behave with what you would get. Like if you did, how we did on the previous page, if you do a Punnett square and you put these gametes on the side of your Punnett square, this is where those gametes come from. This is why we get 4 different kinds. We get this kind, these two kinds if the chromosomes line up this way. We get these two kinds if the chromosomes line up the other way. The fact that these chromosomes are lining up in different ways, what do we call that? Independent assortment. The fact that we get either an A, big A, or a little a in each gamete, we never get two A's in here, that's segregation of alleles. Same with B's. You get either a big B or a little b. You never get both in the same gamete. Another thing to keep in mind is that this is diploid, because there's 4 chromosomes up there, and each of these cells is haploid. One way to represent diploid is 2N, N. There's a total of 4 chromosomes here and 2 chromosomes here in each cell. And how do we know that? Because we count them. 1, 2. And then let's go up to this guy. How many chromosomes are in there? Let's go with another crazy color. I don't have any good colors. And we'll highlight it. 1, 2, 3, 4. And then after meiosis 1, there's 2 chromosomes. 1, 2, 3, 4. After meiosis 1, we have 2 chromosomes. All right, the only thing I guess I didn't label, so we labeled sister chromatids, we labeled homologous chromosomes. Big A and little a are alleles, and the position on the chromosome is the locus. And the plural of locus is loci. So we're only talking about technically 2 loci here. The A locus and the B locus are the positions of those respective genes. All right, so I don't think I want to put too many more scribbles on this page. So look at this, print out, I guess I could give you a blank copy of the quiz if you want to practice some more. And we'll leave it at that for now. Any questions, come see me.